to piece together lots of little pieces of information today. So if I'm bouncing around between my presentation and we're going to watch a couple of videos, so make sure that you stop me if you need some more assistance on this chapter, because this is a pretty important chapter. But the one benefit of this chapter is that we will be covering a lot of the material in lecture. So that will help us put some of this into practice so that it makes sense to us. All right, so the first thing to talk about is kind of the purpose of this chapter is to look at what microbes need to survive, how they interact with each other, and kind of their optimum conditions for growth. And then the next chapter, we talk about all the ways we can stop them from doing that. So this is kind of their chance to shine, and then we'll kind of take over. Okay, so this image is just pulled straight from your book, but it's kind of nice because it shows all of the different ways that microbes, all the different requirements that microbes need to survive. It seems like, right, with the virus and with bacteria everywhere, that these organisms just have a great time and that they have no trouble surviving. They've been around for millions of years, so therefore it must be easy for them. And that isn't true. It's just that they are very good at reproducing, so that anytime conditions are a little bit favorable, they go gangbusters so that they can then have enough of themselves that's kind of how they can survive. Because it really does take quite a bit to allow them to grow well. And so you had a long list in your notes for these. So as we go through today, we'll be talking about most of these, but just make sure that you are clear on those lists. In other words, how do they like each of those criteria? So for example, the nutrient source, the energy source, the temperature, gas content, all of that. I will say one very general trend is that it tends to be like this on a curve. It's just a typical bell curve. And what I mean by that is most organisms are going to like this part of the curve, meaning that they're going to like the median comfortable temperature. They're going to need the proper amounts of nutrients. They're going to need the proper pH. But there are some oddballs that can then survive in these places that are too high or too low than what most of the other ones would survive in. Okay? And if you are a microbe that can take advantage of those locations, say for example on this temperature scale, if most things survive right there in the normal body temperature range, as far as pathogens go, if you are a microbe that can survive in somebody that has a fever, or in a type of reptile maybe that is a little bit colder, then you have a place to live where other microbes can't live, and that can be beneficial. Of course, that means that you will be less likely to survive if you have to compete against everybody else who can grow at that average range much more easily. Okay, so there are benefits and drawbacks to being a generalist, meaning you do what most of the others do, and there are also benefits and drawbacks to being a specialist, meaning you have your own little tiny niche that you can survive in. Okay, so in talking a little bit about nutrition, some of those nutrition requirements are very similar to what we learned about in anatomy. And what I mean by that is things like essential nutrients and growth factors. Those are things that our body needs help producing. And so we have to be able to, and there's a little difference. Those terms are not interchangeable. I do want to point that out in case those definitions were a little weird. Essential nutrients are just any chemical, any molecule that the organism needs that it has to get from its environment because it cannot synthesize it, okay? And growth factors are specific factors that are nutrients, but we can also include them, we can include other groups too, like macronutrients and micronutrients, things that we also need. All right, so that was a lot of overlap there, but I want to go to spend some time on this image. This one's not in your book. This was from a different book. So be comfortable with this one. So this is looking at the organisms that are found all over the place. So we're not just talking about microorganisms. This is a much broader category. So if you look at the top here, This is just a graph for energy sources. So what this is talking about is all living things need a source of energy, right? And it can either be chemical energy, like we do, right? We have to eat our chemical food. That's how we get our energy. Other organisms can use light for their energy. And of course, the easiest example of that is plants. But there are bacteria and protists that use light energy as well, right? So we can split everything into those two groups. So let's just take a few minutes and look at each side of this. Another term for an organism that gets its energy from chemicals or eating nutrients or absorbing nutrients is a chemotroph. Okay, a chemotroph. And that just means that this troph word means to eat. So it's literally saying it eats chemicals. Okay, and its energy is going to be some kind of carbon source. Okay, so the one we typically talk about is glucose, right? That's the typical one that we talk about. And so that is this whole category here. This is the glucose eaters. Okay, so we can call them, let me zoom in here so you can see this, although I don't think it zooms you in, so you should zoom in so you can see that better. So the first category is the chemoheterotrophs. Sorry, it's a little blurry now that I'm looking at it. Chemoheterotrophs. And that means that they eat other organisms. Hetero means different. So they are chemoheterotrophs. They eat different organisms. Or if you want to break it down like this, pencil back. They are eaters of different chemicals. Okay? So we can use examples that we already know about, like all animals. And so this would include the helminths that we're talking about in this class, the worms. Also the protists and the fungi. And bacteria can do this too. Not, bacteria, we forget about them being eaters because they don't have mouths or anything like that. But they absorb their nutrients and they're absorbing a carbon source. Okay. By the way, this final electron acceptor, we're not going to focus too much on this. This is part of their energy system, but we don't have to worry about that. So anyway, animals, fungus, protozoa, bacteria. So this is a lot of what we're going to be talking about. But it's not all bacteria. I just want to point to you real quick that we do see other bacteria over here on this side. So we talk about them all over the place. Okay. But anyway, let's get back to our right here. Okay, so this one is just looking at a different species of bacteria and a different species of bacteria. Okay, so that one that I circled is really the one to focus on. Okay, if we then look at the next category here, so this, remember, we were at carbon source, and we said, what about those things that eat glucose? We call them chemoheterotrophs, and all of our microbes that we talk about will be that type. But we can also have others that are CO2 eaters. Okay, so instead of glucose, we're going to use CO2 as our energy source. And that's really different. I am not talking about photosynthetic things like trees that use CO2 as part of their, that's their carbon source. It's not their energy source. Okay, do you get the difference there? That's really important. CO2 to a tree or to algae is their carbon source, not their energy source. But for this little weird group of chemoautotrophs, these are rare. These chemoautotrophs, this means eating chemicals by myself. So that means eating this CO2 because it is a very simplistic molecule. It's not a big organic molecule that something else created. This is CO2. And so, like I said, this is rare. There aren't too many of these. There are quite a few out there, but we're not going to focus too much on the pathogen. There aren't really pathogenic members here because these live in very strange places. So these are hydrogen, sulfur, iron, nitrogen, and carbon monoxide oxidizing bacteria. So these are things that live in very strange places, like Yellowstone, not Yellowstone. Yellowstone in all of those hydrothermal vents. Okay, I'm going to erase all those marks because that's getting a little messy. But so far we've focused on these chemoautotrophs and these chemoheterotrophs. Okay, so now we're going to go to the other side of the chart and focus on these now. So we have these using light as their primary source. And so we call them phototrophs, which means eaters of light. And so they have their light as their energy source, and then they use organic compounds to build their bodies up, or they use CO2 to build their bodies up. Okay, so the here, we'll go to the left first. So the photoheterotrophs, again, these are very weird ones. These are not going to be any that we focus on in class because they're in a strange group. But green non-sulfur bacteria, purple non-sulfur bacteria. Okay, photoheterotrophs. And then on the other side, photoautotrophs. And again, we don't have to worry about this too much. So here's our photosynthetic plants, algae, and cyanobacteria. That's a very common big group. And the anoxic photosynthetic bacteria, the ones that don't live where there's oxygen, the green and the purple bacteria. Okay, so that's a lot to kind of take in, but what I really want you guys to focus on are that these levels, okay? Chemical energy, and the groups that we need to understand are the chemoheterotrophs and the rare chemoautotrophs. And then for light energy, we have the photoheterotrophs. They eat different organic molecules, or they use different organic molecules. And these ones that use the photoautotrophs that use carbon dioxide, these are the plants that we think of. Okay, so of the things that you already knew about before walking into this class, this would be the plants, this would be the animals, and some bacteria, and the bacteria are kind of all over the place. Okay, so I hope that helps you relate some of that to what you already know in comparing some of those different types of organisms. I do want to bring up this for you because I think I do think, well, I don't know that I need to bring it up. If you have your notes in front of you, I'll just bring it up in case you don't have them. I'm just worried it's not going to work like it didn't the other day. Okay, looks like it worked. All right, so this is that list that I was telling you about that deals with this issue, and it's probably even more easy to look at it here. So we have the autotrophs, the self-feeders is what that means, and heterotrophs, the other feeders. And then it gives you the, all of the autotrophs, the energy source is going to be sunlight, except for those that use these deep sea chemicals, okay, in the deep sea vents. So that's the big difference between the two of those. And they both use CO2 as their carbon source with all those weird other chemicals here that make the chemoautotrophs different. On the heterotroph side of things, this is now going to, I would like for you to know, let's see. This is specific to the fungi. So that's something else that you should put in that list. Okay, so for the chemoheterotrophs, these are going to use organic carbon as their carbon source and organic carbon as their energy source, okay? So they're doing cellular respiration after they eat other organisms. So that term that I was trying to help you understand on the other page, eating the chemicals from others, chemohetero, different troph. Okay, and so fungi do this by being saprobes. That's the term that we talked about with external digestion, where they create an enzyme, exude it from their body, that digests outside, and then they suck it back up. We've got a little picture of that in a second. And a symbiotic microbe will live off of the molecules that another organism creates. Okay. And then the photoheterotrophs can use either sunlight or organic matter as their energy source. They can switch back and forth. So this one is almost the photo part and the hetero part. You could almost do it like this, photo / hetero, because they have the option of being either of those kinds of eaters. They can be a phototroph or they can be a heterotroph. They choose based on what it's like around them at the time. Okay. So what that looks like, if you are a saprobe, those are the fungi. This image from your book shows that process of, here is the little microbe. It could be a fungal cell, it could be a bacteria that can do this. And out here is the blue little organic matter that they're trying to eat. So they, okay, so they will make those enzymes and then secrete them out. That's what the little red dots are. It breaks down the molecules and then they suck them back up. And you know how much I love these two equations, so you should definitely know these for the test. Okay, remember with these, the top one is photosynthesis, and you're basing it off of that one molecule of glucose. So you just put a 6 in front of everything else. 6 carbon dioxides, 6 waters, and 6 oxygens. And then cellular respiration is you just take what is at the end of photosynthesis and put it at the beginning of cellular respiration. And you get out what that's at the beginning of photosynthesis at the end here. And energy, of course. Okay, any questions on those? Because that's like 2 pages worth of your notes that I just went through pretty quickly. 1.5 pages anyway. Any questions on those? Okay, so let's talk a little bit about, let me see where that is in your notes. Yeah, okay, I'm going to slip this in there. So these terms in your notes that the parasite, opportunistic pathogen, pathogen, obligate facultative parasite, and ecto, endo, and intracellular parasites is all review from what we have talked about in the previous chapters. So I'm not going to go over those again, but you know, it's a very quick definition for each of those, so I don't think we need to study those longer. Okay, so yeah, I am going to go into this temperature now. All right, so sorry that's so small. If you need to zoom in on that, you should be able to do that on your end. Okay, so what we're looking at here is a topic that is very important for you to understand with microbes. So what we're looking at are the growth requirements, the temperature tolerance of different microbes. And frequently we'll talk about bacteria in these ways, but it can apply to any of the microbes. And so on the left side, we have the rate of growth. How do they grow? When do they grow the most, in other words? Okay, so they've labeled this green one for us. This green one are the mesophiles, and those are the ones that grow best in us. Okay, and that's something you should know. Mesophiles grow best in us, our body temperature, essentially. So what this is showing us is that they can survive down to temperatures of 10 Celsius. That's their minimum, and they can survive up to temperatures of what, like 45, 50 Celsius. Okay, so they can survive there, but they grow the most. You'll be most likely to find them in this optimum range, okay, which is right at body temperature. Okay, so the mesophiles grow best there. And then you can see how they change after this. The psychrotrophs are like it a little colder, and the psychrophiles, remember phile means love, So the psychrophiles love it colder. And then going the other direction, thermophiles love warmer temperatures, and extreme thermophiles extremely love temperatures, higher temperatures. Okay, so those go up really, really far. That's what you would find in those thermal vents in Yellowstone. Okay, in hot springs, these are the organisms that can survive there. So again, I want to point out that if you are a microbe and you are an extreme thermophile, you can live in a place where very few other critters can live. Okay, maybe a couple of these thermophiles might be hanging around, but they'll never outcompete the extreme thermophiles in this little range right here. Okay, so the downside of that, of being an extreme thermophile, is if you are removed from your environment, you aren't going to do very well. You'll die. You can't survive it, which is weird for us to think about, right? We would think like, whoa, gosh, they love those extreme temperatures, but they'd probably be okay at body temperature. They don't. They are equipped to deal with that heat and process it in a certain way, so they really do die out at lower temperatures. Okay, so which of these would you rather be? Which of these has the widest temperature tolerance? Which one can survive in the biggest range of temperatures? Those extreme thermophiles, right? They go all the way from like 65-ish to 130 Celsius. That's a big temperature range, but it's all very, very hot. Let me tell you what that 70, I'll choose 75. So 75 C to F. Quick calculation, 167 degrees is their low range. Okay, so 167 degrees is this 70 Fahrenheit is comparable to 75 C. Okay, so quite hot. It's just, you know, nothing else is going to be living here, so they really have it made. But let's leave that one out of the equation for a little while. Which of these other ones is then the most wide-ranging? The green one, right? It has the widest spread. The other ones are more of a narrow spread. So what does that mean for us? It means that a lot of the pathogens that infect us grow best at our body temperature and can survive higher temperatures, or in other words, can survive a little bit higher for a short time, like if you have a fever, and can also survive a little bit colder, like if they are being sneezed through the air, the cold air, on a water droplet and land on somebody else, then they can infect that person because that short-term coldness won't affect them too much because they are okay down to this 15 C. So let's see, what is 15 C? 59 Fahrenheit. Okay, so that's, 60 degrees. It was warmer than that today. I hope you got outside a little bit. All right, any questions on the temperature? That's a pretty important topic, and we will do a couple of little temperature checks in lab. Okay, let's see. Are there any questions on the terms obligate anaerobe, facultative anaerobe, and microaerophile? Let me see if I can find a chart for those real quick. I'm not finding one real quick. That's okay. Those ones are pretty straightforward. Usually students don't have much trouble with those terms. Just make sure that you can keep them clear in your mind. So when we say facultative, it means can do one way or the other way. If we say obligate, it means you must do it that way. And aerobe means that you need oxygen. Anaerobe means that you don't. Okay? And so we can have the same chart with those categorizations too, meaning there's going to be a little bit of overlap between microaerophiles and facultative anaerobes, okay, but not an overlap. They do need some oxygen, so we won't overlap with the true anaerobes, okay? All right. So let's look at, let me fix this spacing real quick. Okay, so on this ecology section for microbes, we have these different criteria. This is what we would call them. And then these pluses and minuses are referring to how one affects the other. Okay, so I'm going to make sure that you understand what those pluses, minuses, and zeros mean. So in obligate mutualism, they must live together and they benefit from doing so. So the two pluses mean one of the organisms receives a benefit, is happy, and the other one also receives a benefit and is also happy. Okay, so this is like if you live with a friend and your friend loves to cook but you love to do dishes, you will make a very good pair. Okay? A non-obligate mutualism means that you are better off living together, but you don't have to live together. Okay? So in other words, yes, the person who cooks can survive without a dishwasher. They could use disposables. And the person who likes to wash dishes, I guess, could just dirty them just for the fun of it. Okay, my analogy is starting to fall apart a little bit. But they can live separately, but they live better together. Okay? In obligate mutualism, they can't survive without each other. An example of this obligate mutualism is lichen. And so lichen are those little flat, I'll share my screen so we can go back and forth between these, if it works. Okay. That's what I think I need to do is, yeah, okay, that worked. All right, so this is lichen, and lichen are a symbiosis between algae and between fungus and bacteria. Let me show you a little. We'll watch a YouTube video that shows how the molecules move between the two. Whoops.
To understand what lichens are.
Okay. All right, so I'm not going to have you watch the whole video. There's some pictures of lichens. And what I really want you to see, I'm getting there, is this is how closely the two live together. So this is a little green algae in the middle, and these little strings around it are the fungal hyphae. And so they live that closely together, really tight neighbors, and they provide nutrients to each other. So what the algae gets out of the relationship is a safe place that is moist, and then it photosynthesizes, the algae photosynthesizes, and shares the nutrients with the fungus that's protecting it. Okay, and they can, if they try hard, grow them separately in a lab, but it takes a lot of work. They really do need to have each other to survive. So this is showing the algae in green and the fungus in black and white. So this is all that a lichen is, are these two living in such close connection that you can't tell them apart. It's just showing the different zones of them, of the fungus. But if we go back to a very typical example, so that's what this is. These little round leaves are that fungus sandwiching the algae on the inside, or cyanobacteria. Okay, so told you I'd be skipping around a lot. Sorry, I hope it's not too disorienting. Okay, we were looking at the notes, sorry. I should have just stayed on that screen. Okay, so back to this one. A little bit bigger. All right, so that was obligate mutualism. Non-obligate mutualism means that they're better together, but they don't have to be. So an example might be something like, this is one we haven't talked much about, but it's a really interesting example. So, oh, and what was the name? There's a really good documentary. I'll have to post it for you guys. Anyway, it's trees have fungus that interact with their roots. It's called mycorrhizae fungi. And it forms just as tight of a connection as the fungi and bacteria that we just saw in the lichen. But in this case, the fungi helps the tree absorb more nutrients than it would otherwise be able to do. And in return, the tree gives the fungi sugars. They can live apart, but they do better when they're living together. Okay, they can show a tree that has no mycorrhizae and it's just shorter, looks a little thinner than the trees that do have the mycorrhizae. Okay, and the next example, this one is commensalism, and one of them receives a benefit while the other is unchanged. And it's kind of hard to talk about these in bacterial worlds, so I'm going to give you a large animal example. But say, for example, it is a sheep, and it's walking through a field and eating, doing whatever it does, whatever sheep do, walking around, eating the grass, drinking, whatever. And a bird perches on the top of the sheep so that it can see where the insects are. Okay, so in that example, the bird received the benefit because it was able to use the sheep as a perch so it could see its food better. And the sheep doesn't care. It's not going to bother the sheep at all. Okay, so in the microbial world, this would be where one organism receives a benefit from another, but the second one doesn't care either way. So maybe it could be something like a fungus that secretes a molecule for some reason, and it just happens to help a bacteria. The fungus gets nothing out of it. Okay? And that's kind of the example that I gave you here with the syntrophy. Okay? Or a dung beetle, right? Eats the dung of cattle. The cattle don't care if the dung beetle eats the dung, but the dung beetle benefits. Oh, and by the way, you can really use this as an insult to somebody. Instead of calling them a bad name, you could call them, man, talk about that person using syntrophy. It's basically saying you eat somebody else's waste. Kind of funny. And nobody will know what you're talking about, so then you won't hurt anybody's feelings. Unless they're in this class too. Anyway, let's go to parasitism. That one's pretty easy to understand. One benefits because it's eating off of the tissues of the other. So the other one that's being parasitized, of course, suffers. That's parasitism. And so that's what we would see with the helminths, the worms that would survive inside of us. We don't usually use the term as much for things like bacteria, but you could. Technically, it is a true thing to use the term parasitism about bacteria. The last one is amensalism, where one produces toxins to harm the other. I don't think that's supposed to be a plus minus. That's supposed to be a neutral minus. Hold on, let me verify that. That doesn't look right. Yeah, that's supposed to be a minus. So fix that in your notes, okay? This is supposed to be a neutral minus. Oops. Sorry, I did not notice that before. A neutral minus. So one produces toxin that harms the other, but the original didn't necessarily do that just to harm the other, okay? So there's like a no issue, but what the like no benefit or harm to one, while the other one is suffering because of it. Okay. All right, so any questions on those? So can you tell me when you look at those, which ones are symbiotic and non-symbiotic? Symbiotic means that they work together. And so typically we use the terms mutualism for those. You could argue that commensalism is that, but because one has no change, you know, it's not being harmed. So you can consider the commensalism and the syntrophy to also be symbiotic. But the parasitism and amensalism are definitely non-symbiotic. One of them's being harmed, so therefore it's not good. Okay, so, oh, sorry, were you guys on that? No, okay. So now we're going to go back to the presentation. I am in a class. Sorry about that interruption. Okay, back to the presentation. So now, oh, I know what I wanted to do before we go on to biofilms. I wanted to show you a few videos. Sorry, I forgot about that. Jumping around, trying to hit all the topics on this big chapter. Okay, so yeah, you can see that. All right, so this is just showing how bacteria divide. I just, as we move into how quickly bacteria can divide, I just want to show you this. This is just going to run a little ticker across the bottom of the screen. So it's sped up, but this is just watching them and taking successive pictures through a microscope. 1 hour, 20 minutes. 1 hour, 40 minutes. And you can notice, like, once we get to this 2-hour mark, we're growing much bigger, much more quickly because we're going so rapidly. All right, so, you know, that is exponential growth, and we are going to learn about that today too. And so after 24 hours, I don't think they give the actual amount, but you can figure that out. It's a lot. It's like billions of billions. All right, this one is pretty cool. We're not going to watch all of it, but this is mold instead of bacteria, so fungus. And again, this is time lapse, so it's sped up. This is fungus growing on an agar surface, like you guys saw. Those are the little fruiting bodies that are popping up. Conidia. And then there's one, show you this one real quick here. You can see the Conidia individually on some of those. And then when it starts to turn darker, that's because all the little spores are being released. And so they're just settling, some of them are settling back down to where they are. And one more. This is slime mold. And you may have seen this before. This will grow in your like mulch or leaf litter, usually more mulch, or even on a recycling, like a compost bin. And this is unicellular. And this is a time lapse, so it's sped up. But if you take like a little stick and touch this, it'll like flow out as though it's bleeding or something. But these are all individual organisms, but they work together to cover an area. And I don't know if you've ever seen, if they have this in this video, they can learn how to run a race. This is kind of like what they're doing. Do you see how they kind of like pulse when they get close to something? Like the other cells that are part of this colony will respond because they can communicate to each other. So they're like, ooh, a food source, let's grow really fast here. And before all of the other ones get there, so they're communicating. See how that kind of looks like it's pulsing? It acts as though it's a living tissue, even though it's individual little organisms. Okay, anyway, so those were just some cool videos that I found about. Oops, this one. All right, now on to biofilms. So the biofilms come in as a discussion of those commensalism topics. And we've talked a little bit about what biofilms are, but this is just a refresher on that then. So this is taking us step by step, and it starts off here to the left, where we see these little microbes that are going to be the first colonizers. So they swim around, and when they have found a suitable substance, they start to settle down. So they literally just stop being mobile and decide that this is a good place and we're going to stay here. And then they start to exude a matrix of molecules that's going to help hold them down so that they can't easily be wiped off or eaten off or attacked by other microbes. And remember that other species can come in. This one doesn't show the other species, but we can have other species come in. And then we can have those quorum sensing steps. Remember that? Where the whole group, when it knows it's big enough, can then decide to do a new response, create new molecules, or interact with the rest of the biofilm in a certain way. But it won't do it until there's enough of a quorum there, which means enough of the group for it to be successful. Okay? It would almost be like if you wanted to cause trouble and you had to have enough people with you for it to succeed, and if you don't, it fails. Like you shouldn't even try unless you know you're going to be successful. That's how these bacteria are. They know when they have enough, and then they can be activated. And this is just showing some of those changes that can occur after that. Those are some of the things that they can do after quorum sensing. Okay, like, and this one example, making a new protein or making some new enzymes that can digest food particles. All right, so remember I said last time that we would talk a little bit more about how we can replicate bacterial cells. Since this is the growth chapter, this is where we hit some of that. And we'll do again after we understand a little bit more about genetics. But here's our first introduction to that bacterial replication of binary fission. So this is our parent cell. We still call them a parent cell, just like we do in mitosis inside of our cells. And the parent cell is going to start to elongate. That's what we see starting to happen here. And then replicate the bacteria chromosome. And this is occurring at those 2 replication forks I've mentioned just a tiny bit of, but we'll learn more about that when we get to genetics. It's just another introduction to that. So it replicates the bacterial DNA. And then each one goes in its own separate category, and they break off, and they have 2 identical cells. So unless there's been some weird mutation that's unexpected, all of the cells in a colony will be genetically identical in bacteria. Okay, so what we're leading up to here is an understanding then of exponential growth, and that's kind of what we were watching in those videos. So this is what exponential growth looks like on a little chart like this. So we're looking at the number of cells that we initially start with. So 1 cell is all that we need to start binary fission going. We do not, this is not sexual reproduction, we just need 1 cell that divides into 2. Okay, 2 divide into 2 more each, so that's 4, 8, 16, 32, and so on. Okay, so this is after 5 generations, we have 32 individual little bacteria. Okay, and so we are increasing them. We are basically doubling everything that we have. That's why we're always going up by 2. Okay, so this 2 times 10 to the 5th, because we're on the 5th generation. And from every 1 that we have, we can make a result of 2. Okay, so the reason that, the reason I bring up this image right here is because you need to understand it's 1 cell dividing into 2. Just like mitosis, it's not 1 cell giving birth. Okay, it is 1 cell dividing into 2. And that's how we get these numbers across the bottom, this exponential value. So that exponential growth looks like this if we put it on a chart. Okay, so as time goes by, we have a certain number of microbes. And so I just want to make sure that you understand what we're doing here. At the 500, if we take that 500 and double it, we get 1,000, right? If we take that 1,000 and double it, we have 2,000. If we take that 2,000 and double it, we have 4,000. So within those generations, if let's just say that we, I'm just going to start here at time 1. I'm just going to say that this is some 1 time. And then this is time 2. We doubled the number of organisms, right? And then time 3. And then time 4. Okay, my lines aren't perfectly straight, but what happened on this axis? We covered about this much of this axis, but we covered about this much of this axis. Oh, a little too high. About that much of that axis. So what we need to understand about exponential growth, and this is really important, as we double, the amount of time that it takes to grow the overall population is logarithmic growth. It goes up by huge amounts. Okay? And so one way to understand that is, let's see if I can find that real quick. I'll show you this video. Okay. This should work. Yep, okay, that should work.
The story
about lily pad, where an invasive species of lily pad gets on a lake, and within 20 days could infect the entire lake. So I thought that's a pretty interesting visualization. And I thought I would show a little bit about how that works. So this is Lake Dirty Hands. And can you see the invasive lily pad? Let's zoom in a little further. How about a little further now? Can you see it? See the green dot right there? Well, there it is. So on day one, this lily pad can reproduce itself every single day on that. So day 2, there's 2. Day 3, there's 4, 8, and so on. Day 7, day 8, day 9, day 10. So it looks like we have a pretty big problem. But if we zoom back out, right there is all that we have covered in the lake. So now let's look at day 11, day 12, day 13, day 14, day 15. So you could be on this lake and not even realize that there is a problem with this invasive species. Now let's do day 16, day 17, day 18, day 19. So one day before the entire lake is infested with this lily pad is only halfway on that. Here's day 20. I don't mean to share this to cause panic.
Okay, so what do you guys think about that? Another interesting thing about that exponential growth, I'll give you one more example. Let me look it up real quick. I don't want you guys to see the answer. This one went straight to the answer, but I'm going to give you another stumper for exponential growth. Okay, so I'm just going to talk through this one. I don't want to give you the answers to it. So if we have a piece of paper and we fold it in, let's see if this will be it. This will give me the actual measurements I need. Okay, so if you fold a piece of paper in half and then fold it again, it's 4 folds as the original one, and then that gets you to about 1 centimeter, okay? And so, oh, it's not exactly right. Let me find that. Hold on. Okay, this will work. I'm going to snip this and show you guys this piece of the information. Okay, so if we fold, that's really big. If we fold a piece of paper, oh, that came out so big, how strange. 1 time, that's the number of folds, it will be 0.2 millimeters. So this is going from very small. So if we fold it 4 times, it gets to be 1.6 millimeters. And remember, there are 10 millimeters in a centimeter. So we're still talking about only as thick as 4 sheets of paper. That's how this folding is working. Okay, well, 8 sheets of paper. Okay, because we've taken one piece and we fold it in half once to get us to 0.2, fold it in half twice to get to 0.4, 3 times 0.8, 4 times 1.6. So how big do you think it will be if we fold it 42 times? 42 times. We folded it 4 times, and see, we're doubling it each time, right? We took a piece of paper and folded it in half. So going from 0.1 and doubling that is 0.2. Doubling that is 0.4. Doubling that is 0.8. Doubling that is 0.6, right? So how big will it be by the time we get to 42 folds? Anybody want to take a guess? How many people think it will be more than I am tall, like more than 5 feet tall? Do you guys think less than 5 feet tall? Give me an answer. Type some in the chat or just talk out. I'm afraid that I've lost y'all. More or less than 5 feet tall? That is a common guess. Anybody else? 4. Anybody else out there? Check to make sure people are speaking with me. I know I'm kind of scattered today. I'm sorry. I'm just trying to pull some pieces together for this. Okay, so it's more. So we just got more. Is it more than a house is tall? I think it's more or less than a house is tall. Let me show you a little bit more of the data. Go ahead and guess how long it is. Really important is how big is it? Measuring them like this. My voice is probably so sharing the video. Let me be back again. All right, there's more data because we stopped at the 4 folds of 1.6, went up to 3, 6, 12, 25, 51, 102, 204. So if we're changing that to meters, we're just going to move the decimal points a couple spots to the left. So what's going to happen after 20? We're going to be at 200,000 millimeters. So if we are at, let's do it. I'll do it this way. So that's 104 meters right there. Instead of millimeters, I'll just move it 3 decimal points. Now we're at meters. 104 meters. That is like after this high, that's like the height of a plane roughly that a small plane would fly. So when we get to 40, let me just stick the rest of this. So here was our 21 at the top with 200,000. So that's 200 meters, 400 meters, 800 meters, on and on and on, all the way up to this one. Here is our meter point. So 439,804,651 meters, which is actually to the moon. So do you get what that means? If you could fold a piece of paper 42 times, you would get to the moon. That is a legitimate calculation, a legitimate measurement. It's called the paper fold to the moon exercise. And it's all just based off of starting that one tiny little bit. At what point do you feel like we really took a turn for the huge? Maybe somewhere in here, right? When we're going from let's see, this is 1 million meters. Now we're at 3 million meters, 6.8 million meters, 13 million meters, 27 million, 54 million, 109 million, 219 million, 439 million. So it climbs exponentially fast. And that is what bacteria can do because they demonstrate exponential growth. Because every time they make more of themselves, there's twice as many as there were before. Okay? And so the rate of change increases rapidly. And that's what we were getting from this exponential growth curve. Okay? So where we had this 500, that was like the turning point on this one, or maybe 1,000. Because up until this point, it took all this time to get there. But then as soon as we hit that kind of turning point, it's just the amount that makes it really increase quickly. 1,000, 2,000, 4,000. Okay? The human growth curve, by the way, the human population growth looks very similar to that, except that we are starting just a little bit to reach the top of this curve. Because what you expect to happen next is this. Because nothing can increase exponentially forever. Here it is inside of test tubes. That's what this one was showing. There's none, and then you put a couple in, and then there's 4, and then there's so on and so on. Why didn't this one turn to 8? Because maybe one of them died. Okay, it's not going to be a perfect exponential growth curve because of all of this. So this is our big lead up, and this is probably the last thing I'm going to talk about today. Okay, you will need to be able to create this graph and label it and explain what is going on in each of these steps. This is a bacterial growth curve, and if you don't know it well, I'm going to make you keep redoing it because this is kind of the crux of the whole course. You need to understand that this is how bacteria live, but it's also how other organisms live too. So overall, before we start explaining the big details of it, we're going to show that this is the number of cells. This is the number of cells are going to increase, and then something's going to stop them from just continuing exponential growth forever, right? If this was exponential growth, it would go like this, and it would just keep going straight up as the numbers increase. Okay, but are things that are going to stop them from doing that. And those are going to be factors that are out of the bacteria's control, such as the opposite of all of those things that we talked about at the beginning. So the opposite of all of these, when the temperature becomes incorrect, or when they run out of space, or they run out of nutrients, or they change the pH. But most often, and the easiest thing to kind of understand is that they're running out of space, nutrients, or there's too much waste. Okay? In other words, they're just getting overcrowded. There's too many right there, and some are going to have to die off. Okay, so that's kind of the basics of what's happening overall. So now let's take this step by step and make sure we understand this. All right, so in the first phase, this is the lag phase at the very beginning here. And we're looking at the number of viable cells on the left-hand side. And this is a logarithmic scale. Okay, and we will do an exercise in lab. I'm going to add a little thing in there about logarithmic scales when we come back together. But anyway, it means it's increasing by a factor. But anyway, let's just pay attention to the trend of the graph. So during the lag phase, the cells aren't growing rapidly yet, even though conditions are ideal, because they're kind of gearing up to divide rapidly. So individual cells might be growing. They might be internally making the nutrients that they need to grow bigger. So the lag phase is a preparing phase. We should not see many dead cells at all. They should all be live, but they are getting ready to start taking off. And then during the exponential growth phase, they are growing exponentially, and everything is nice for them. Otherwise, they wouldn't be growing right now. So they have optimum conditions for growth, and very few of them are going to be dying because there's no reason to. Everything's great. But then something is going to change. And like I said, it's probably going to be a lack of space, a lack of nutrients, or too many wastes that are causing them to decline. So for a while, they'll be able to maintain their numbers. And in this stationary phase, this is probably the one that students get wrong the most. It's not as though everything is holding still. It is just that the number of new cells being created equals the number of old cells that are dying. Okay, so one way to look at this is to write it as birth equals death. Okay, what we were doing before this was birth is greater than death. There's more births than there are deaths. Sorry, that should be a D. Okay, and so in that stationary phase, don't tell me everything's just living fine. There are still some that are being created, and there are some that are dying. Okay. And then eventually it just becomes too much for the organisms to tolerate, and then we go through the death phase where now we have fewer births than deaths, so death is greater now. Okay, and how far that goes, it just kind of depends. If they can level out again, because waste was the issue, and with fewer of them there's less waste, then they might continue on with just a gentle up and down variation. But if it was that they ran out of nutrients, unless there's new nutrients coming in, there's not going to be any more growth because they've used up the nutrients. So they may go all the way down to 0 if they've used up all the nutrients. Okay, so that's why we don't give a real final end to that curve. It could depend. So hopefully you can see that any of those factors can cause this same type of curve, but usually it's going to be one of those 3 factors that I mentioned. And within a biofilm, they may be able to mitigate some of this because of that quorum sensing. So for example, if the nutrients become too low, then maybe some of the bacteria in the biofilm could take advantage of other nutrients that the other ones can't, and then share those nutrients. Okay, so they could reach a stationary phase and stay in that stationary phase for a really long time. Okay, any questions on that one? Okay, all right, that's all I'm going to cover from chapter 7. So unless there are any other questions that you guys have, you can take off just a few minutes early.